Our company is founded now almost 20 years ago in Frankfurt by molecular biologists, and we are experts for next-generation sequencing library preparation and methods around this and bioinformatics. And we produce kits for the next-generation library preparation, especially of RNA, and we also provide, of course, services and the bioinformatics. So here's an overview about the different technologies. Mainly, as I said, RNA is our main focus. We have these very sensitive methods that I will show later. Of course, we also provide genomics analysis in there, especially the bioinformatics is our main, let's say, expertise. Then epigenomic information, like the methylation of the DNA or the RNA, is something we provide, and also the interpretation of the data, and then, of course, also the microbiome analysis. And as I said, we produce these kits, but also, of course, the service. We started out as a service company and then realized that we have so much knowledge about how to produce the libraries in a very efficient way, so that we said, okay, let's make kits from that. Okay, why RNA? Yes, for you, it's probably very obvious that RNA is a very interesting level of information that should be analyzed and used more. But in the clinics currently, RNA is playing a very minor role only for clinical decision support. So everything is based on the mutations, or maybe on if RNA is used, then it is looked at, for example, if there are fusions between genes. So more the qualitative information of the RNA is used, not so much the quantitative information. Before few quantitative RNA data is used, the exception is breast cancer, where this is still from the days when microarrays were big, when a lot of data was produced using microarrays, and then Oncotype was the first company or the first test that predicted if adjuvant therapy was necessary after a breast cancer or breast tumor surgery. And then the other companies came up with other tests, but all of them are based on RNA level, and this is based on the analysis of FFPE tissue. Then very recently, I just found this. There's a new test which is FDA approved, which is a test for RNA that looks at RNA level expression in stool. And the question is, why are there so few RNA-based tests out there? And first of all, I think one thing is probably it's very, very complex. Okay, maybe not really an argument why it's not used for a biomarker, but still I think this is interesting to know that how many different complex processes are happening. We have this called transcriptional pre-mRNA processing. There's alternative splicing. There's the mRNA translation efficiency, which plays a role in the localization of the RNA. Of course, the stability, protein binding, methylation. There's RNA editing, post-transcriptional changes of the sequences, etc. tRNA usages, and then, as I mentioned, these different classes: microRNAs, long-encoding RNAs, circular RNAs, etc. And we only usually, if we analyze RNA, we only look at a fraction of this, mostly mRNA or microRNA. So all the other RNAs we don't look at. And then we have these technical problems with RNA analysis. There are many, many different ways to analyze the RNA, many different protocols. It starts with the isolation. Then what kind of poly-A capturing do you do? Which method do you use? Do you use RNA depletion for the analysis? How was the library preparation done? If you look at NGS, do you use a strand-specific protocol, or do you use which polymerase do you use? Temperature, hexamers, etc. And then, of course, the bioinformatics workflow, which is probably the easiest to be standardized. And then, of course, we have the maybe larger problem, which is that RNA degrades, of course, very fast, as we all know. So it's not maybe the ideal medium to look at for an IVD. And the other problem is that we have differences of the degradation. So if we have one, let's say we want to analyze 100% of our patients, but only 70% have the quality of the patients is good enough to analyze it. So you probably don't like this kind of IVD test. And then also you have differences in the degradation. And maybe one sample has very good quality and the next sample has bad quality. And that again makes it more difficult to make such a test. Okay. So the requirements then for such an RNA-based test would be, as I think, a very good solid expression measurement method that also can deal with partially degraded RNA, especially FFPE-derived RNAs. Especially in the tumor field, we deal always with FFPE-derived RNA still at the time. It should also be cost-efficient because maybe if we look at maybe the single-cell world, the costs are, I think, still prohibitive for standard diagnostics. And it should be easy to use. And, of course, you can predict that. I want to show now our solution, which we think fulfills these approaches or these requirements, which we call MACE-seq, which stands for Massive Analysis of cDNA. And now we just look at the molecular background of this. So we have here the RNA molecules or population of RNA molecules that have been translated into cDNA. So we have the green transcript, let's say, 3 times, and the red and blue transcript once. And then after cDNA production, it looks like this. With a double strand, we then perform a random fragmentation, very much like an RNA-seq. So there was this paper pasted in. And after this random fragmentation, the RNA is, we only keep the 3' ends of the molecules, so only this part. So we don't sequence the rest, which is why this is more stable. We don't have a strong difference between degraded or partially degraded RNA or differently degraded RNAs. And then we sequence this part, and we keep those tags basically. So for each transcript molecule, we end up with one single read, very much like in a PCR where you don't sequence the entire molecule, but just take a certain region in the qPCR, and then you measure the level of your gene expression by using this qPCR code. So this is the same. So we can count then these different tags, and then what is nice is that we can reduce the required requirement of the sequencing depth, so we reduce it to about 5 million reads because we have only around one-tenth of the sequencing molecules, of the molecules that we need to sequence. So that makes it much cheaper, and it's very nice for high throughput sequencing or large numbers of samples. Just cost savings always. Okay, now if we look at the two techniques head to head, we have here the RNA-seq on the left side, and I depicted the small RNA with this little square here, and on the right side we have the MACE-seq. There's a fragmentation step, and in the end we keep the 3' fragment. Now what is a very big difference, and this is why this is working so well for degraded material, is that on the if you use hexamers, only a part of the RNA is transformed into the cDNA, because the hexamers will not bind at the very end of the RNA, but somewhere, and so we will never get the full fragment, full size fragment. Whereas here with the MACE-seq, we do not use hexamers, but we will then obtain the entire fragment of this 3', and this is longer than these fragments. So all of these short fragments we lose, is including the small RNA. The other thing is we can go down with the input amount down to single cell amounts now, and so we end up with longer reads, and we have this 1 read 1 transcript. Okay, so this is the workflow. Here it takes about 3.5 hours to generate the libraries you need to extract RNA, and then NGS and then the bioinformatics. You can do this either internally or we can do it for you. There was just recently this paper came out, and compared it with the RNA-seq, and they found this much better. The protein coding the protein coding genes were better identified basically with the MACE approach. So our vision is to use more quantitative data for the therapeutic decision support, and the way to do this is first of all, what is already out there is quite amazing. I mean, I think the last maybe 30 years there have been RNA data has been collected with the microarrays and then with the NGS, of course. So currently there are thousands and thousands of data sets available, bringing together feature with a gene expression profile. And then of course we can see also now that there are more and more scores with clinical relevance generated. If you look for these two words, then we see a strong increase, almost exponential, for this connection between RNA profiles and clinical information. And but however, this is not really available for anybody because there's no such a collection or database like what we are used from DNA, where you have a certain mutation that you know if you have this mutation, that this and this happens, so this is known. So what we tried to do was to make a collection of this clinically relevant information, getting the phenotype and the scores that are connected with this phenotype in a big database. And so we then generated a classifier using machine learning approaches to obtain, let's say, for the different characteristics, a classifier, and then we could obtain, which I here not depicted as this barcode, and then if we find the same barcode in the example, we make a prediction of what the characteristics of the sample may be that have clinical relevance. So we did this in a project funded by EU with the samples from colon cancer, and here we see, I think these are 120 samples from colon cancer patients, and we obtained these different features from each of them, which are written on the right side. I don't know why this is not working anymore. So some of them we had to black out, and for example, there's this responder to fluorouracil on the side here, and this patient has a profile that looks like one that we found that is very likely to be well responding to it, whereas another patient, probably this one, has a very low score, so it's probably not as likely to be well responding. So we have all these different features, and we now provide this in a report, and then together with Pablo Ganesa, we also tested some of this hypothesis. Of course, all of these need to be tested and looked in detail. And this is a drug prediction, so we have these organoids, or the group in Spain generated these organoids. We obtained the RNA, made the classifier, and made a drug prediction, and then checked if this drug was true, and it was also checked with other drugs. And this was then tested with different drugs, and here we have the different scores. For example, this PF29984 was supposed to be working very well. COBA was another drug that's supposed to be working well in one PDO, whereas it shouldn't be working well in the other PDO. Of course, we all know about TME. We heard a lot about TME, so I won't say a lecture on this, of course. But what is interesting is that, of course, the most data about TME is currently known or is collected from bulk RNA-seq data. Now, of course, in the recent years, single-cell sequencing is used, and but I think that single-cell for, as I mentioned before, for therapeutic decision support is still too far away, too expensive, too difficult. So if it was possible to use bulk RNA-seq to make clinical predictions on the TME, that would be great. And there are several works of this, and one of them is this Cassandra program, where they have also simulated, like we heard before, and also used real data to derive single-cell composition of a TME by RNA-seq, bulk RNA-seq. I think this is a great example of how this would be done. Another example that we did which included the TME, was to examine if we can find biomarkers that predict the patient response towards immune checkpoint inhibition therapies. This was done in collaboration with Helios Clinics, which is a large clinical chain in Germany, and the Charité. So we obtained, I think, 80 samples from responders and non-responders to immune checkpoint inhibition, PD-L1 and CTLA-4. targeting drugs, and then we came up with the score. We tried to, of course, we have this problem of overfitting. It's not such a large data set, but a lot of data, so this is always dangerous. We tried to use a very robust score for that, and currently it looks quite nice, but of course there needs to be more validation to be done on this score. So what it does, it basically collects these features. If these features are present, then they are added up. If we then end up with, let's say, if more than 17 features are present, then we can make sure, or we can say, okay, the patient should receive the drug, or if it's below this score, then should not receive the drug. And with this score, we could increase the prediction likelihood, at least if it's validated better, then we know more about it. But these are the current numbers with the data that we found in the internet and our own data. Just a few last words about RNA data for liquid biopsies. Again, here we see a very strong increase about RNA biomarkers in this field, so there's something coming up for sure. However, again, no FDA-approved tests are available. One of the big problems here was the contamination with leukocytes, and this is again how the samples are taken is very important. When is the centrifugation done? How much time is between the sample draw and the centrifugation passing? And many details need to be taken care of. I would like to show you our new RNA, small RNA analysis. On the left side, we see again the RNA-seq, and now we came up with a method which is a little bit based on the Maze-seq method. Okay, and again, we have our RNA molecules, and basically, again, we can see on the left side the hexamer-based RNA transcription, but on the right side, okay, on the right side, you see in pink that the entire molecules, all the fragments are basically transferred into the library. And this is a big difference because for the first time, we can analyze the mRNA and the small RNA simultaneously without losing much information. And again, what's also nice, the input amounts are very low. So we can go down to single cell amounts, which is perfect for example, if you work with EVs from liquid biopsies. So the amounts are very low and the sensitivity is very high. And if we now look at the number of molecules, because for biomarkers, of course, the main thing is how many different transcript species do you capture. And we see on in blue, the former small RNA-seq protocol, which is based on ligation, which really captures only small RNA molecules down up to about 80 base pairs. And on in orange, you see what we can obtain with the pan RNA-seq approach. These are the same samples, the same tissue, the same samples that were split in two. We then performed our already very good small RNA-seq protocol, which is also used for a lot of clinical or biomarker studies already because it has a good quality already. But now we could beat this by far with this pan RNA-seq. So what are these molecules that we capture in addition? Again, in red here, the pan RNA-seq, and in blue, the transcripts from the classic small RNA-seq approach. And here we see that we capture much more long non-coding RNAs, and all of the samples had more pRNA transcripts, which are also longer than the protein-coding RNAs. We can capture much better. And if we look at the microRNAs, there are some cases where we had more microRNAs captured with the old method because it's focusing really on the small RNA, so maybe that is the reason. But in 80% of the cases, we even had more microRNAs captured with this method. Okay, last words about our bioinformatics. Since we are in a very, let's say, in a shark tank, a lot of competition, etc., so we must focus on the high-quality analysis, of course, and we put a lot of effort in a pipeline to make a very comprehensive analysis of RNA. So we offer this, of course, to anybody who wants to send us his RNA data, and then we perform a very comprehensive pipeline consisting of course, the mapping and the feature quantification, and then we go through all these different databases, provide the data for the, let's say, meta information about the disease gene networks, G profiler, wikibarbers, breakpoint, etc. And what we also add, in addition to this, is we look at alternative polyadenylation, which is a feature of RNAs that is important for the stability of the RNA. We also look at xenoread, for example, from viruses or other contaminants that may be in your samples. We do HLA typing, mutation calling, little splice size detection. Just wanted to mention this because there's a lot of times there's a lack of bioinformatics competencies, and RNA can be easily sequenced and very cheap, but then often the bioinformatics is not keeping the pace. So with this, I'd like to thank you for your attention, and all my colleagues and the grant agencies that are listed here, and especially to the group of Papa Conessa. We worked